problem. Why is plant growth affected by gravitropism?
Hypothesis. If a plant is placed on its side in a dark location, then there will be changes in curvature of the stem because negative gravitropism will occur. This movement is due to the large oxygen concentration that accumulates at the bottom of the stem, which stimulates cell elongation in the stem.
The independent variable for this experiment would be the position of the plant within the box, with one plant being placed perpendicular to the ground and the other plant being placed parallel to the ground. The dependent variable for this experiment would be the observed change in curvature for both the control group and the experimental group. The control for this experiment would be the plant placed upright and erect, perpendicular to the ground, so that it acts as a basis for comparison to the experimental group, which will be placed parallel to the ground on its side. Some constants for this experiment would be the soil used, the species of plant, the amount of water used, the location of the experiment, the humidity, the temperature in the room, and the environment used, which in this case refers to that each plant has no light, since we don't want phototropism to possibly affect our data.
Materials needed for this experiment are 2 of the same potted plant, 2 boxes that are big enough to fit the plant, a pair of scissors, a ruler, tape, and 3 cloths big enough to cover the box. The procedures are as follows. Number 1. Make sure that the potted plant is saturated with water. If needed, cut the branches and leaves in order to be equal to one another. Label one of the plant experimental and the other control. Measure the tallest stem for both plants and record it. 2. Place the experimental plant on its side inside the box and put a piece of cloth around the pot to limit the pot from moving around. Then take the control plant and place it right side up inside the box. 3. Close the boxes and secure each box with tape. Then cover each box with a piece of cloth. 4. Place both boxes in a dark location like a closet to prevent the light from reaching the plant. 5. Leave the box in the closet for 10 days for best results. 6. After 10 days, take out both of the boxes from the closet, remove the plant from the box, and record the change in curvature of the experimental plant in comparison to the control. Now, here's the data that we collected, a comparison between day 1 and day 10. Before each plant was put into their experimental habitat, each plant appeared erect and straight. After a week of being placed in their separate environment, the experimental group was noticeably more bent near the base of the stem when compared to the control group. The control group appeared most similar to before the experiment took place. The difference of curvature between day 1 and 10 for the control is 1 degree off, while the experimental has a change of 65 degrees compared to day 1.
Analysis. To find change in length, you will essentially measure the height of the plant from the base all the way to the tip of the plant. You will do this for both the control group and the experimental group on day 1 and on day 10. To find change in angle, you will use a protractor to find the angle the plant makes with a flat surface at the base of the stem, and then find the angle the tip of the plant makes with the same flat surface. Next, subtract the base angle from the tip angle to get change in angle. You'll do this again for both the control group and the experimental group on day 1 and day 10. To find curvature, you'll divide the change in angle by the change in length for both the experimental group and the control group. To find change in curvature, you'll subtract the curvature on day 1 from the final curvature on day 10 for the control group and the experimental group respectively. Trends and data. In the data table, some trends can be established across both groups. When the plant was placed on its side parallel to the ground, The curvature changed much faster than the curvature for the plant placed erect and perpendicular to the ground. Also, it is noted that the curvature of the plant did increase for both, just at very different rates. It's also worth noting that both plants exhibited negative gravitropism, since both plants grew against the force of gravity. The change in length for the experimental group was 2.1 centimeters greater than the change in length for the control group. The difference in the change in angle from day 1 to day 10 was much greater for the experimental group, as its change in angle changed 65 degrees, while the difference in the change in angle for the control group was merely 2 degrees.
The problem was, why is plant growth affected by gravitropism? To start off, we must understand what gravitropism is and how it occurs. Gravitropism is a turning or growth movement by a plant of fungus in response to gravity. The hormones responsible for gravitropism in plants are auxins. Auxins are hormones that regulate plant growth. The plant stem is growing in the negative direction away from gravity, and this growth is promoted by the function of the auxins, which stimulate elongation of the stem cells. After conducting this experiment, we found that our hypothesis that if plants were placed in different positions / experimental habitat, in this case in a dark environment on its side, then they will still respond to gravity because gravity is a powerful force, and negative tropism will still apply to this situation. And in each case, the plants grew upward, demonstrating the effects of gravity whether the plant was placed straight up or slanted to the side. This is shown in our pictures of the plants and data processing. Overall, the hypothesis was supported.
Post-lab questions. Identify the hormone responsible for the behavior you measured. The hormones responsible for gravitropism in plants are auxins. Auxins are hormones that regulate plant growth. The plant stem is growing in the negative direction, away from gravity, and this growth is promoted by the function of the auxins, which stimulate elongation of the stem cells. During gravitropism, there is an increase in auxin concentration on the lower side of the stem and roots of the plant. Due to this high auxin concentration, the roots cease to grow longer while the stem grows upward. Cytokinin is a hormone that promotes cell division, or cytokinesis, in plants. This is a secondary hormone that will regulate the transport of auxins within the plant. Question 2. Identify the structure and cell type responsible for secreting this hormone and describe how the cells responded to accomplish the behavior you measured. Auxins are produced in the stem, buds, and root tips of a plant. The stem, buds, and root tips are made up of plant cells. Within the root tips are statocytes, cells that contain statoliths, an organelle that stores starch. When influenced by gravity, the statoliths pile at the bottom of the cell, which promotes a new growth pattern and ultimately makes the roots grow downward, positive gravitropism, and the stem upward, negative gravitropism. Question 3. Connection of your work to the podcast Smarty Plants. In the podcast Smarty Plants, the topic discussed was whether plants could learn. There was an example of an experiment where a fan was placed near a plant, then a blue light was shown on the plant from one direction. Due to phototropism, the plants began to lean toward the light. The order of the fan, then the light, was placed on the plant for 3 days. The plants were then only given the fan and no light and began to lean toward the fan in anticipation of the light shining. In this gravitropism experiment, the experimental plant was placed on its side in a dark location. The results were that the plant began to grow in response to negative gravitropism, away from gravity, and not in response to light. The conditioned plants mentioned in the podcast began to lean toward the fan due to the expectation of light, not in response to it. In both cases, light was absent and did not have effect on plant growth. Question 4. Connection of your work to the podcast From Tree to Shiny Tree. In the podcast From Tree to Shiny Tree, trees are sharing nutrients between entwined root systems that are infiltrated by fungi. The trees can send signals to the fungi and send signals to other trees through the fungi system. In this experiment, the plant can sense the direction of gravity and grow based off the sediment of statoliths within the sensory statocyte cell. This gives off a biochemical signal, and auxins are then transported to this new growth pattern in high concentration.